Okay, great, excellent. So today we will continue our discussion about the biological physics. And then especially today we will talk about some basic quantitative calculations that are useful for all of us to have a quantitative view of the biology. Okay, and then we will talk about a few basic laws that we will find useful in the future class. And if we have time, we will start to talk about the fundamental 4 building blocks of the life. Okay. And here I have only one announcement today. So for the reading, besides the book I mentioned that the basics of life uploaded in the Canvas, I also put another paper. That's also a very interesting paper. Talk about the brief history of the Hodgkin-Huxley model. That is the model we briefly talked about, the action potential electric signals in the neuron membrane. Okay, we will talk more about the electric signals in the later half of the class, and then we will show how to better measure and even manipulate those electric signals. Okay, that we will discuss that once you have learned all the important biological knowledge and the physical principles. Then we will get to that topic. But it is good that you it will be fine that you read this brief paper here. Okay, good. So let's start today's lecture. So in last lecture, we talked about the laws, theories, and equations in the biology, right? So we talked about the law of evolution. So that was developed by Dr. Charles Darwin, right? So we all know this law now, but at that time, at his time, it is really a big breakthrough in the world. Okay, and then based on this law, people knows that life is not static; it's actually dynamic, and it has a certain direction to go. It will evolve and then become more and more efficient or effective, right? Another law we're talking about is central dogma of the molecular and the cell biology. That basically says the information transformation from the gene to the RNA and then to the protein. Okay, so and also this including the duplication of the gene. Okay, I mentioned about those black arrows in last lecture. That's important concepts: transcription, translation, and replication. But I didn't talk about this red arrow go backwards from the RNA to DNA. Do you guys know what does this mean? How can the RNA become DNA? Do you know about that?
Reverse transcription.
Exactly. It's called reverse transcription. So this always happens in the virus. Okay, virus can inject the RNA into a cell or into a bacteria, and then that can be converted into the DNA. Okay. Now, do you know when you look at this central dogma? Do you think there can be other arrows to be added into this picture? Is it possible from the protein go back to the RNA? Is that possible? Have you thought about this?
I don't think so.
Why? Why?
Because you need like the opposite of a tRNA.
Opposite tRNA. Can you make the answer to be more complete?
Yeah, I guess I'm basing it off of, I know there's like new sequencing technologies to sequence protein, and I believe it's like the only method now is to sequence protein, and then to replicate it, you can make synthetic DNA or RNA rather than going in reverse.
Excellent. So what you said is the statement of current new technology and what they can do, but you actually didn't answer why protein cannot go back to RNA. You didn't answer that, right? You did not answer that. What's the theory behind it? That you didn't answer, right? The reason is from RNA to the protein, there are many options. From one RNA, there could be multiple types of the protein because protein has some redundancy here. Same coding of the RNA can give multiple different amino acids. So then if you have many solutions here, from those solutions, you actually cannot go reverse. You cannot go exactly back to the same RNA. That is not a one-to-one map. That's the reason. Okay, but it will be important when you look at this diagram. You ask these questions: Can other new arrows be added into this central dogma? Okay, because many Nobel laureate, Nobel Prize, they get many Nobel laureate, they receive the Nobel Prize. It's because they find those arrows. Each arrow here is one Nobel Prize. If you can find another one here, you will get a Nobel Prize, right? So, and this actually is drawn by the human being. We don't know whether anything actually at some point you find some exceptions maybe there, right? So it's always good to think about this.
I have a question.
Please.
So why wouldn't there also be an arrow maybe from the protein to the DNA, because protein can bind to DNA and affect whether it's
yes,
able to be transcribed?
Excellent question. So that is the protein work with DNA together to replicate the DNA or give rise to the RNA. Here, those arrow means from one material converted to another material. You are talking about the material work together, give another material. That actually is implicated here. From DNA to from the reactor. Reactor is DNA. Reactor material is DNA, and then protein is helping that, and then give the product. Product is another DNA, right? So that is actually implied here. Good question. It's clear now, right?
Yes.
Excellent. Good. So in the class, please join the discussion and ask those crazy, bold questions. That's the point of a special topic class, because we want to have more discussion, more ideas to flow, right? The idea should flow like a river, like a very rapid river, right? Should be like that way. Good. Okay. And then we talk about the equation. One equation in the biology, like on the membrane. So this is a membrane. And then, because of the concentration difference between intracellular domain and extracellular domain, the membrane is not inert. They actually are electrically charged. The membrane, transmembrane, they have an electrical voltage there. Okay. Do you guys see my mouse?
Yes.
Yeah. Excellent. Just in case, because in the campus recently there are sometimes has this interruption of the internet, right? So if that happens and we get lost in the internet, if the class the time is over, I will be happy to record a new lecture online, and then you can read that. You can watch that. Okay. So don't get panic if the internet get interrupted. Okay. But I will ask you from time to time whether you can hear me or not. So just let me know yes, and then I can continue. Okay. Good. So because those membranes are electrically charged, and then on the membrane there are many ion channels and many electrical pumps. Those electrical conducible channels, proteins, pumps, actually they have different dynamics. Some can be opened quicker, some can be opened slower, some can close quicker, some can close slower. So they have different dynamics. Okay, so they are not only conformationally different, but also functionally or behaviorally different. That gives rise to the unique hallmark of the electrical dynamics on the membrane. So this equation actually just talk about the static voltage of the membrane. Just because of the internal and external ion difference, you can measure the voltage on the membrane. But if you consider the different kinetics of the proteins embedded in the membrane, then you can model the membrane like a circuit, okay? Like a circuit containing the capacitance. Capacitance majorly is the membrane, and then it also contains the charge, the battery inside. There are other pumps, right? The motor proteins pumps, and also they have the adjustable resistant inside, and this resistant is representing the closed ion channel. When the ion channel is open, then resistance decrease, right? And so by modeling this, by considering all those factors, then you can understand why there is an action potential. Why there's an action potential on the membrane. This membrane is actually not all type of cell. It's only the cell that is excitable cell. Means that the cell contains all those electrical conducible mixed proteins. Okay? Some other cells do not have those proteins, then they do not have action potential, even they have the membrane voltage. Okay? Then by knowing the properties of those proteins, we can actually write an equation to describe this model. And then surprisingly, this equation actually can represent most excitable membranes. Okay, here in this equation is a very elegant equation. This equation, they only consider two types of proteins embedded on the membrane. One is the sodium ion channel, one is the potassium ion channel. Those are all selective ion channel. Only let the sodium in, only let the potassium in. But they have different dynamics. One is faster, one is slower. And then because they are mismatched, so when they open and close, they will collectively form this kind of electrical pulse. Okay, and then those pulse can repeat for many times. So this is actually the beauty of integration of physics, that it can help us to model the biological property and then abstract it into a concise equation to describe it. And also is the beauty of the biology. Here on the membrane, because of evolution, the cell contains all those ion channel, those proteins that allows the membrane not only behave like an inert lipid layer by layer, but also display electrical signal. And then this signal can propagate for long distance to communicate with the cells that are far away, right? That give rise to the activity of our whole brain, And then our brain, the reason our brain can think, can perceive information, can compute information, can store information. And also the reason we can have emotion is all because of those electrical signal there. And those electrical signal are because of those membrane and proteins there. And then those membrane and proteins are actually because of their unique physical, chemical elements there, right? So when I just mentioned, we will talk about the 4 basic building blocks of the life. Here actually 2 types. One is a protein, one is lipid. So this already talk about the 2 different unique building blocks, right? Another 2, one is the DNA, RNA, and the nucleic acid, and the third one is sugar. The fourth one is sugar. So these are 4 building blocks of the life. Here, using 2 building blocks, you can form the language in the neuron system, right? So this is actually the combination, the integrated beauty between the physics here and the biology here. So yeah, this model is called a HH model. You can read that reading materials I put it there, and these two these two scientists, they win the Nobel Prize, and then they did an experiment. They derived this model by doing the experiment on the lobster, in the ocean. The lobster has very large neuron compared to human. Lobster and as well as octopus. Okay, those deeper ocean animals, and they tested their neurons and then find those models, and then those models actually also apply to the human neuron. Okay, so right now this is the outline overview of the class of our class. Now we finished the introduction, and we are stepping into some basics of the life. This section mainly is used to keep all of us on the same page. So all of us, if you don't know biology before, as I promised, we will teach those in the class, and then we also provide the reference in the canvas. You can read those materials so that all of you can know all the terminologies, all the concepts, biological concepts, and biophysical concepts. Then we can go deeper into the mathematics, different types of mathematics, and the physical tools such as imaging tools, and also the biomechanics. Okay, so let's focus on this the second block. Okay, so when I talk about the theory of the cell, there actually is another theory people always think is fundamental. That is called the cell theory. They call it the cell theory. That actually means all the living matter, living system consist either of one cell, such as bacteria, or multiple cells, such as us, human. Okay, so they are not continuously connected. They are always when you look into the living system, they're always composed of cells, many cells. Okay, the first person identified this theory is actually called Robert Hooke. Do you guys hear me? Do you guys see my mouse?
Yeah.
Yeah, excellent. So the first person is called Robert Hooke. He actually is a very productive scientist and engineer. Okay, he also developed the Hooke's law in the mechanics, in the elasticity. If you learned the continuum mechanics and the elasticity before, you will know what is Hooke's law. Okay, and then use Hooke's. At his time, he actually used the spring, single 1D spring, but then that actually can be expanded to multiple dimensions. It later on can be expanded to the big matrix to calculate the Hooke's the principle of the Hooke's law in the 3D composite materials. Okay. So besides mechanics, Robert Hooke also is an expert in the optics. So here actually is the first compound microscope constructed by Robert Hooke. This compound microscope contains many lenses, and collectively those lenses magnify the view of the subject. Okay, and then from this view here, sample plan, to the view of the to the plan of the experimental's eyes. Okay, to the retina. Okay, and then in their retina, they find if they take a piece of cork tree, put that on the sample plan, they find there are many small segments on the slides. Okay, and he called this each segment, this black one, each one, we call it a cell, each a cell. That's the first time the cell, this word is coined. Okay, and then later on people find this actually is correct. Everyone is a single independent unit, and all the cell actually they have almost identical components inside, and then this what they find in the plants actually can also be found in the animal, in the different types of animal, and then then overall they call those small elements called a cell. Okay, and then the cell theory what it means is it says the cells are the fundamental unit of the life. All the organisms are composed of the cell, and all cells actually are coming from the pre-existing cells. So cell can divide and the cell can differentiate, especially for the stem cell. Okay, and any multi-cell organism, multi-cell system actually coming from one cell is a fertilized egg. This fertilized egg actually is coming from the fusion of two cells. One is the egg, one is the sperm, and then they become fertilized. Then they will mix the genomes and then start to divide. And then during the time, at the beginning time of the small embryo, there are many stem cells there. And then stem cell can differentiate. They feel the environment, differentiate, and then divide. Okay, so eventually they give rise to the whole multi-cell organism. So here actually some examples. Here you can see on the top row, these three are the single cell level for three different multicellular systems like sea urchin, mouse, seaweed. They all coming from the single cell at first, and then at the first glance, single cell level they are looking very similar. They are only the cell, but then as they grow, they will grow into very different systems, right? That's because the gene inside, the DNA inside all three are dramatically different. The sequence is very different. So that's the cell theory. One question people always ask is, if you look at the single cell, but there are also single entity such as virus, okay, like a COVID-19. The virus is also look like a single cell, right? Those are all virus, different types of virus. So the one question is, are those single virus also the cell, also the alive, also are they regarded as the living system or not? Do you guys know that?
They're not, because they're only composed of RNA, right?
They can contain DNA as well. Virus can contain DNA. They can have DNA and RNA together. Yeah.
Is it no because
yeah, go ahead.
No, because they can't reproduce on their own. They need like a host.
Exactly. Yeah, that's the reason. So virus, actually people disagree. Most of the people disagree. I think more than 90% of people think virus is not the living entity, because they have DNA, sometimes they have RNA. Though their DNA and RNA cannot replicate by itself inside this small entity. They need to find a host, either a bacteria or a cell, and then inject those DNA into the cell and the bacteria. And then within the cells, within those hosts' body, the DNA of the virus can be replicated, because they take advantage of the motor proteins and the protein complexes in those hosts' body. And then once the virus's DNA get replicated in the host body, they will self-assemble into many new viruses. Okay, that's how the virus proliferate. So by itself, they cannot pass information to the next generation. So that's why they are not regarded as the living system. Okay. So that's good. That's good. So it's very important to clarify this. and this property that the virus can take advantage of hosts' machinery inside, actually has always been used in research lab. Even in our lab, we actually generate a virus. We can program the DNA in the virus. We can make the DNA there to be the gene we want, and then we put those virus into the cell, and then kill the virus quickly. Then the DNA inside the virus is transferred into the cell we want, like the neuron. into the cell, and then those gene will be translated to the protein, and then those protein is used to study those neurons. So we actually generate many virus in the lab. So we take advantage of the property that the virus is very, very effective in transfer their genetic information to the host. Okay, so people like to always do this. This is called bio-inspired engineering. Right? So people like to do this. So this is one example. Okay, so if you look at the, as we said, talk about this cell theory. Okay, it will be important we look into an even bigger picture, even bigger picture. So if we look at all the living systems, except the virus, okay, if we look at all the living systems on the Earth, we can actually group them into 3 types. One is called, from left to right, one is called bacteria. So this is the one, bacteria. Another group is called archaea. Okay, I have a picture of archaea in the next slides. They look very, very similar, bacteria and archaea. Third one is called eukaryote. We are eukaryote, okay? Human are eukaryote, and the plants and the other and the plants here, plants, animal, we are animal, right? And now the fungi, those are all belonging to the eukaryote, okay? So on the Earth, there are 3 types called 3 kingdoms of the life. They all coming from one ancestor that is hypothesized. They're all coming from one ancestor. And then gradually through the evolution, they become 3 kingdoms. Now, in these 3 kingdoms, initially actually when people study this, they think there are 2 kingdoms. One kingdom, they actually didn't separate bacteria, archaea from the eukaryote, from us. Okay, it didn't separate that. But then there's a scientist called Carl Woese. He actually is the modern, it's called a modern Charles Darwin. He compared the ribosome RNA. Remember ribosome, I talked about ribosome. That's a very, very high, is a hybrid unit, right? Ribosome that contains the unit of genetic information and the protein information, right? So, but there's a type of RNA that can directly talk to a ribosome called RNA. So Carl compared the sequence of the ribosome RNA of bacteria and eukaryote. He found that actually these two, the RNA are actually very different. They are not belonging to the same type of kingdom. Okay, so that's because of him. Then right now people believe there are 3 branches. When the life evolve, there are 3 branches. Okay, and then the bacteria and the archaea actually they are called prokaryote. Okay, we are called eukaryote. Okay, and here is one typical prokaryote cell. Okay, so here is the bacteria, and can you guys hear me?
Yep.
Yeah. Okay, so this entity actually contains a very different cell membrane and the genetic information. It's much more simpler than the mammalian human cells. So it contains the DNA is called a nucleoid. It's actually a single chromosome, not like a human. Human have 40 plus chromosomes. Okay, so for the prokaryote, they have only one single chromosome, and that chromosome is called the blue one. It's not enclosed in the membrane into a nucleus. It's actually diffused in the cytoplasm. Okay, and it contains very rigid cell wall. So for this prokaryote cells, they actually contain the cell wall, and usually two layers. Inside is called a cytoplasmic membrane. Okay, and the outside is called a cell wall, and this cell wall actually is quite rigid. So that's why this cell can maintain this this unique cylindrical shaped structure, right? Because the outside cell wall is very rigid, that can maintain the bending, so that the whole cell is not like a sphere. Okay. And for the prokaryotic cells, as we said, we have the bacteria and the archaea, right? So this is the bacteria. So the one example is the E. coli, and it can be found everywhere, bacteria. Another one is the archaea. That is the actually looks very similar to the bacteria, but this entity, this creature, actually can grow in very harsh environment, such as in the Yellowstone. Okay? So they can sustain very extreme environment, very extreme heat, pressure, acid, salt, and extreme gas. They don't need too much oxygen. Okay, so these two entities are very different, and even you see they look much simpler than the human being, right? They look much simpler than human being. They actually contain very useful machinery in their body. These two, actually, there are many Nobel Prize. They become Nobel Prize because people find important principles and important machineries in the bacteria and in the archaea. Okay, one Nobel Prize is called PCR. In the lab right now, it's very common in the biological lab. People do the PCR. You know what is PCR? Polymerized chain reaction. You know that? Do you guys know? Yeah. So PCR is actually a method. It's a technique. That technique, maybe sometime I can show you the diagram of how the PCR is used, or sometime I can show you in my lab. So PCR is a method that people use to duplicate the DNA in the lab. If you want more DNA, then you can use PCR to duplicate, and it can be any piece of DNA. If you want, you can duplicate that piece. Okay. So the inventor, this is just a story. The inventor of the PCR, that inventor win the Nobel Prize already. He come up with that idea when he was driving on the highway. Okay, on the highway he was driving, and then he saw the rod come together, merge together, and then separate apart. Then he's a molecular biologist. He combined with his knowledge on the DNA duplication. Then he find that in the lab we can actually replicate. We can actually develop a tool, develop a machine to also induce the replication and the expansion of the DNA. That actually is very desirable. Okay, but to realize his idea, there's one step. To make those DNA to be expanded, he needs to, in a controllable way, he needs to put the DNA together and separate DNA apart at the desired time. To do that, he used the thermal heater, used the high temperature. So at the high temperature, DNA can separate. Okay, and then when temperature cools down, DNA can bond together. But if he high up the temperature, make the temperature too high, then he couldn't do DNA polymerization. That needs the DNA polymerizer protein to work. Okay, so that's the barrier he encountered that couldn't get his idea realized. Luckily, he find that because those archaea can sustain very extreme condition in the nature, they can sustain in this very hot spring in the Yellowstone. Then he find that there are certain DNA polymerizer or motoprotein in those archaea actually can sustain those high temperature and still can work. Then he extracted those proteins and then put it into his experiment and make the PCR to be feasible. Okay, so this is one example that those small entity looks simple, the small entity, they actually are very useful if you use them well. There are other examples, like currently very promising gene editing tools. They're actually coming, CRISPR gene editing tools. They're actually coming from the bacteria. They're coming from bacteria, okay? And also the currently very promising optogenetics, membrane voltage imaging, those high technologies, they actually become possible due to the understanding of the membrane proteins of the archaea. Okay, all those are coming from those simple organisms. So when we learn them, when we understand those simple organisms well, we actually can develop many useful technologies. Here's one more example. Let's talk about the evolution timeline of synthetic biology. So synthetic biology is a very important field. Okay, so here from the 1962 on, all the way, every year, every several years, there's a milestone, big discoveries. Okay, and then go all the way up to 2016. Okay, and among all those discoveries, the bacteria, the prokaryotic cell, bacteria or archaea, actually play important role here, like these optogenetic tools, CRISPR gene editing, and also the genetic circuits in the cell, okay, developed in the E. coli. Those are all because of those small organisms. Okay. So those are the prokaryotic cells. Now let's look at the eukaryotic cell. There are also many interesting things in the eukaryotic cells. Can you guys hear me?
Yeah.
Yeah, excellent. Very good. So in the eukaryotes, here is one example, just one cartoon of the typical animal cells. Okay, so here they have this is a membrane, and then they contain the mitochondria inside. Mitochondria is the energy factory of the cell, and then they contain the nucleus, and then lysosome. This is the factory that can do the degradation of the waste inside the cell, do the recycling. Okay, and then the endoplasmic reticulum. So this unique structure, brown color, unique structure in the whole cell. This is called endoplasmic reticulum. If we look at the whole cell like a city, then this nucleus is the city hall. Okay, government is there, and then endoplasmic reticulum, ER, is the post office. This place is the post office, because all the RNA coming out from the DNA will meet the ribosome embedded in the endoplasmic reticulum, and then become protein, be translated into the protein, and then those protein will be ER. This ER will put the stamps on each protein, put a small peptide tag to the protein, and then ship out those proteins. And then because of those different stamps, the protein will go to design the location of the cell to perform their function. Okay, so this is the post office. And then here is the Golgi apparatus, very important, Golgi apparatus in the cell. Okay, so on the right-hand side, these are the cancer cells. Those are cancer cells. Here, what I show the picture is the cancer cell. The bottom here is the cancer cell before they become invasive, before they become malignant. On the top, those are circular, rounded, each one is a single cell. Those are single cell. Each single cell actually is a malignant cancer cell. Those rounded cells are ready to invade the patient body. Okay, and then from this non-invasive cancer cell to those invasive cancer cell, this transition process actually is realized by all those organisms, by all those organelles. Okay. And when we talk about the biophysics, actually two types of organelle in the mammalian cells are very important. One is called cytoskeleton. Another one is the membrane. Okay. Cytoskeleton, let's talk about them one by one. Cytoskeleton is like the skeleton in the cell. Okay, here in the sand carton, we add 3 different types of lines there and 3 different color. Those 3 different color represent the 3 different cytoskeleton. They are called, respectively, they are called actin, microtubule, and intermediate filament. 3 types. Okay, those 3 types of cytoskeleton, they actually play really different roles. If you remember the video I showed you in the last week, the video shows the inside of the cell. You actually have already seen 2 types of cytoskeleton. One is actin. Remember the polymerization and depolymerization of really thin filament, right? That is actin. And also another type is the microtubule. Remember that the kinesin work on top of a thick, really thick cylindrical look green pipe, right? That is microtubule. Okay, so these 2 are very common. Third one is called intermediate filament. This one, although it's not very often mentioned by people, but it plays a very critical role in the cell mechanics. These 3 different types of cytoskeleton actually can be distinguished by their diameter, the diameter, cross-section diameter. Actin is the smallest. Actin has the smallest diameter. It's about 7 to 9 nanometer actin. Okay, but in reality, actin actually can bond together. There are many actin can bond together to be a big bundle. Okay, microtubule don't bundle. They actually live by themselves, and the diameter of microtubule is the highest among all the cytoskeleton filaments. It's about 24 nanometer. Intermediate filament is in between. It is about 8 to 12 nanometer. Okay, so those numbers actually tells us how flexible are those filaments inside of the cell. Okay, as you can see, microtubule is the most non-flexible one, right? It's the 6th one. And
are these all made out of the same material?
Not the same material. Excellent question. Not the same material. They are made of the different proteins, and they are made of different protein monomer, and then those monomer connected together, polymerized together to be the entire filament. Okay, those monomer also have different mechanical properties. So the overall filament's mechanical property determined by the material itself, 3 distinct materials, and the geometry, 3 different sizes here. Okay, excellent question. Excellent. And then these 3 cytoskeleton actually play different role. They have very distinct tasks, and that can be seen in the following figure. So in this role. So first one, this one is the actin. Okay, so the green one, actin. You can see actually it's coming, this the nuclear, coming from the nuclear towards many different places. And you can see it's actually quite floppy. It's actually quite floppy. If you look at the microtubule here, the red one is very straight, right? Microtubule is very straight. So because it's very straight, that's why the kinesin, those motor proteins can work on top of it, right? It's very straight. So the distance is optimized. Okay. So for the actin, it's extremely useful for the following multiple cell functions, such as the cell migration, muscle contraction, because actin is associated with the myosin motor protein, right? So they can contract and then have the muscle collectively, they give rise to the muscle contraction. Also the cell adhesion, because those actin actually bound to the actually actin is actin start from the inside of a cell and ends at the membrane, like here in the cell membrane, and then they can bound to the cell adhesion proteins, bound to that, and then adhesion proteins bound to the external environment. Those adhesion protein on the cell surface serves as both sensors and actuators. they can sense the environment, external environment's features, such as the mechanical rigidity, topography, right? Topography, and the friction of the environment. And then those protein will transmit that information to the actin, because they are bounded to the actin. And then actin will transmit those information back to the cell. Then the cell will know what the physical environment look like. And then the nucleus can make the corresponding commands, corresponding response, okay, to make sure they can survive there. Okay? So actin is a very important information transmission structure inside the cell. Okay. Second one is the intermediate filament. This intermediate filament actually is both a structural element, also functional element. In terms of the structure, this intermediate filament actually can anchor to the membrane and then hold the cell, help to hold the cell on the location in the tissue, and then give rise to the cell structure. It can also help the signaling transduction. Mostly, these intermediate filaments are studied in the cancer cell, because when the cancer cell, as I showed in the previous picture, from the cluster of the cell into those rounded single cancer cell, during that process, the amount of intermediate filament actually increase. Those intermediate filaments actually help those cells become more invasive. They can help them to invade, help the cancer cell to pave the way forward to migrate, to invade. Okay, so that's why the intermediate filament is also very important. The last but not least one is the microtubule. Okay, microtubule, of course, it can help shape the cell, but also they can enhance the motor protein transportation to help the material to be transmitted to different locations of the cell, right? And also another important function of the microtubule is it will help the cell to divide into two and then separate into two daughter cells. So microtubule is very important in the life of the cell, and that's why microtubule actually is used as a target in the cancer biology for the cancer drug, because one hallmark of the cancer cell is they can proliferate crazily, they can divide crazily. So people design some drug to inhibit the microtubule in the cancer cell, is to stop the division of the cancer cell. Okay, so this is a very useful target by the pharmacologist. Okay, but all three of them are very useful, and it depends on different types of the cell. They play different weights. Okay, and they all can help all together. They help the entire living system to work. Okay, in the health and in disease. Here's one more picture showing this is a purely active. Okay, this actually is a super resolution picture. They can remember what I said. The actin filament, the diameter is less than 10 nanometer, right? So if you use the optical microscope, you will not be able to see the single actin. Okay, optical microscope has the resolution limitation, half wavelength, right? Half visible light wavelength, about 250 nanometer. Now, but if you use the super resolution, you can see the actin filaments in the nanometer, sub 10 nanometer scale. Okay, here you can see different color, the blue and green and red and yellow. Those different colors represent different height. So this is the XY plane, but towards the screen, outward point out to the screen is Z dimension. So here the color depends on color denotes the Z axis locations. And you see here there are many single filaments, and this cell actually is cancer cell. They are migrating. So when they migrate, you can see those single filaments actually bundled together now. they bundle together and then protrude forward. Because when the actin filament bundle together, they are much more rigid, right? The cross-section area become larger, even they don't change actin protein materials, but the structure determines the rigidity here, right? So here you can see the cell is actively generate those bundles to push forward, right? And then inside the cell, you can also see the actin get connected together to form the network to sustain the location of the cell nucleus. And then collectively, they move the whole cell forward, okay? Here actually going the left bottom, okay? Good. What I just said is the actin filament's structure. But beyond the structure, in fact, within those actin cytoskeleton, there are also mechanical force embedded. Okay, there are also mechanical force embedded. Right now it's 3:52, so I should stop here, and we can discuss this in next lecture. Okay. Good, very good. Any questions?